Hi, my name is Erez Lieberman Aiden, and in this talk I'm going to tell you about how we can map swarms of 3D contacts using DNA sequencing in order to determine how genomes fold in species like the locust. I want to start you off with a simple hypothetical puzzle. Suppose you wanted to understand the relationships in the fictional town of Springfield, whose citizens include this gentleman. Well, there's many approaches that you could use, but here's one. You can start taking photographs of all the citizens and watch them go about their daily business. For instance, here's a citizen eating a donut. Here's another picture of the citizen. They're interacting with a different citizen. Intriguing. We take more photos and more photos and more photos until we start to get a decent data set. And what we can do with this data set is put it into a table. What this table is showing you is the frequency with which we see a pair of citizens in the same photo. It's the same data as the previous slide, just put into the table. And immediately you can see that this donut-eating citizen and this blue-haired citizen seem to be interacting at a very elevated rate and indeed have given rise to a whole family of citizens with an elevated contact frequency. How does that work? Well, we don't necessarily quite know, but it's an intriguing lead that we can follow up on. Turns out that Hi-C allows you to map contacts among genomic loci in much the same fashion. If you're interested whether two genomic loci are nearby in 3D, what you can do is cross-link the DNA, cut it into little bits, fill in the ends of the bits with biotin, ligate those ends together, and then pull the biotin down. What you've done in that process is you've taken bits of DNA that are nearby in 3D and produced chimeric sequences in 1D that can be readily sequenced and used to determine which sequences were nearby in 3D. This allows us to figure out things like how the genome folds. For instance, here's a model that we proposed on the basis of Hi-C data. We said we believe that there's some sort of machine called an extrusion complex that lives in the bowels of the genome. And it's made up of two subunits that land on DNA and then start sliding in opposite directions. But they don't slide forever. Like when I'm driving on the road, I stop when I see a stop sign, but only if that stop sign is pointing towards me. The subunits are the same. When they see a stop sign, turns out it's a particular motif that binds a protein called CTCF. When they see that stop sign, if it's pointing towards them, they'll stop. If it's not pointing towards them, they won't stop. You can see the top subunit has stopped, the bottom subunit hasn't. Oh, and there it goes. Now it's stopped. This is actually a mechanism that forms loops in the human genome. We hypothesized this based on Hi-C data, and it's been subsequently verified using beautiful single molecule experiments as well as a variety of other techniques. One of the other great things about Hi-C data is it makes it really easy to do what the Human Genome Project did for a matter of hundreds of dollars. And you're going to hear a bunch more about that from Olga Dudchenko, who's one of the other PIs at BPRI. Thank you guys so much for your time.